We are live.
Hello and welcome to our 12th annual Rebel Grad Slam 3-minute thesis competition. My name is UNLV and I'm the director of the Graduate Research Pathways. The 3-minute thesis or 3MT competition model was developed at the University of Queensland in Australia in 2008. By 2011, the 3MT competition phenomena had spread around the globe, and now more than 900 universities from more than 85 countries hold these competitions. In 2014, UNLV held its first 3MT, the Rebel Grad Slam, and this year we have 12th annual Research Rumble. The 3MT is a fantastic opportunity for students to learn how to convey who they are and what they study to an audience of folks who are most outside of their field. It is an opportunity to build public speaking and communication skills. It's worth noting that our slammers come from all over campus and from a wide range of disciplines. This week, we have over 100 UNLV graduate and professional students assigned to compete in the preliminary rounds. Each will present their research in 3 minutes or less, using one slide and no props. We will introduce each student and then start the timer. If a student goes over 3 minutes, they will be eliminated from the competition. Now let's turn our attention to the scoring for this research rumble. The judges will be assessing the competitors based on metrics which total 24 possible points in 4 major areas: comprehension, content, engagement, and communication. Each judge will independently score each competitor. Judges will not penalize competitors for unanticipated disruptions that are beyond the slammer's control. At the end of the presentations, we will tally the judges' combined scores for each grad slammer and announce the winners right here live. The top 3 in the session will advance to the semifinals. So without further ado, let me introduce the judges, and then we will get on with our 3MT Research Rumble. We have a set of very distinguished judges in today's session. Judges, please feel free to turn on your camera on briefly to wave hello as I introduce you. I would like to introduce Susie Scarrow, Associate Professor and the Librarian from the College of Urban Affairs.
Hello, everyone.
Dr. Jake Thompson, Associate Vice Provost, Undergraduate Education and High Impact Practices. Dr. Daniel Babb, Professor in Residence, Honors College.
Good morning, everybody.
Dr. Javier Rodriguez, Professor and Executive Associate Dean, School of Life Sciences.
Good morning.
Dr. Kian McMahon, Professor and Graduate Coordinator from the Department of History.
Hi, everybody. Happy to be here. Thank you.
Dr. Yu Ha-Yoon, Assistant Professor of Sport Management.
Good morning, everyone.
Welcome, and thank you for serving on our panel of judges today. So we are ready to begin the competition, and our first presenter is Matthew Caruso.
Just like to remind our judges to please enter ones for Madeline Price in order to advance to the rubric for Matthew. And Matthew can begin whenever you are ready. We'll start the timer as soon as you begin.
Hi, everyone. I'm Matthew. I am an occupational therapy student in my second year of 2027 with my cohort, and I was going to go over my capstone project for sound therapy. Now, as everyone may know, we do have something in our brain called brain waves, which activate at different frequencies during different times of the day, and the different levels that correspond with what we're doing when we first wake up, when we're sleeping, when we're in a stressful situation, or when we're enjoying something of one of our hobbies or occupations that we do enjoy. At the different levels, we have delta, which is when we're in deep sleep, very, very quiet, very calm, nothing's going on, usually no movements of the hands or feet. In theta, we start to wake up. We're usually in a very calm sense, usually when we're eating breakfast or just maintaining any sort of various activities that we tend to enjoy. In the alpha wave, this is when we start to pay attention, such as in class, when we're focusing, and I'll touch on alpha a little bit later, which is the reason why I'm doing the capstone project. In beta, we're usually working on something a little bit more difficult, such as taking a test or working at our job where we need to be able to correspond to different activities happening at different times. And gamma is when we're in high stressful situations, when we're tend to be focusing on more, such as driving or taking a test. Now, during these times, our brain doesn't understand what's going on around us, more what we're focusing on at that current moment in time. Now, I wanted to go back to alpha because when we're in alpha state, usually we're in the middle of class, focusing on what's being taught in front of us. And even, I know I do this too, whenever I'm focusing on some sort of hobby, I'm humming. And during that humming, my frequency is very low because I'm in a flow state when I'm working on something. Now, with sound therapy, what we work on is trying to relax the brain so that it can focus on something specific that we're working on. And where I got the idea for my capstone project was I was a kid with ADHD and I want to go into the pediatric field. And while I'm working with kids, I noticed that they usually tend to have a problem focusing, and especially the ones with ADHD can't sit still. And it's not because that they have any lesser ability to do so, it's because their brain has a difficulty of staying in that low alpha, high alpha, low beta uh, brain frequency. And what's going on with that is their ADHD brain is just all over the place. They're focusing on other things, they're always constantly moving, and they can't sit still. So, with my plan, what I wanted to do was to bring in sound therapy to help those kids that have that learning disability. And by doing that, I would give them headphones to place over their ears while they're doing a worksheet or assignment or taking a test, or even just while they're listening to the class or to the teacher. Oh, shoot. Can't believe that was 3 minutes. Sorry.
Thank you, Matthew. Let's give our judges 60 seconds to score your presentation. Thank you. Our next presenter, Mark Brennan.
As the fresh water of the Mobile River rushes out into the salt water of the Gulf of Mexico, something remarkable happens. This water doesn't collide, it actually blends together, forming a different type of water called brackish water. And what makes this so important is that it actually makes Mobile Bay the home of a lot of different types of wildlife. And what this means is that we have some that live only in that fresh water and others that spend their entire life out in the ocean. But some move in between this gradient of water. And what I've realized is since moving to Las Vegas and my research, that this is actually paralleled by mental health disorders as well, where a disease like PTSD is actually characterized by depressive and anxiety symptoms, resulting in a heterogeneous condition that's actually marked by high rates of treatment resistance because each individual is going to exist in a different presentation along this spectrum. And our current treatments are one size fit all. It's kind of like throwing a hook out into the water and just hoping that something bites. But if you know anything about fishing, it's actually really important to know exactly what you're looking for so you can have the right tools for the job. You need to have the line that's big enough and strong enough to not snap. You got to have weights to get that lure down to where the fish is. And it's got to be something also with the lure that fish wants to eat. And we aim with our research to bring this level of precision to pharmacological treatments of disorders like PTSD. Now, the way that we do this is by defining the spectrum between psychedelic and entactogenic therapies. As currently viewed as separate classes, their overlapping efficacy as well as structural similarities highlight that these conditions or these drugs are better characterized as existing along a spectrum. And what this is going to allow us to do is to have intermediate presentations of disorders matched by hybrid compounds that exist in the same parallel spectrum so that we can give treatments to these individuals that are personalized to their individual presentation of these heterogeneous disorders, allowing us to more effectively treat those that suffer from these disorders because it's not just on the shore or all the way out deep in the ocean, but it's actually all the waters in between where we have the healing that is needed.
Thank you, Mark. Now we will give the judges time to score. Thank you. Thank you so much. Our next presenter, Elmira Ebadi.
Imagine being a child in Las Vegas, where summer days climb past 110 degrees and a school playground feels more like a frying pan than a field of play. Now imagine trying to learn math or read a story in a classroom that overheats before lunch. Teachers here in Clark County describe students as tired, sleepy, miserable, and not learning. This isn't just discomfort, it's a barrier to education. Hello, I'm Elmira, a Master of Architecture student, and my thesis asks a simple but powerful question. Can the shape of a school courtyard help children learn better in desert cities? Courtyards are one of the oldest design strategies for hot climates, from ancient Persian homes to Spanish mansions, because they use shade, airflow, and light to create comfort. But modern schools often treat courtyards as leftover space. What if we could use science to rediscover how courtyard proportions like height, width, orientation affect heat, daylight, and energy? I am building a digital model of a K-8 school and testing hundreds of courtyard shapes, like stretching and rotating a cube, to see which form works best under the Las Vegas sun. Using advanced software and a genetic algorithm, a kind of artificial intelligence inspired by evolution, the computer learns which courtyards perform best and optimizes new generations of design. Each option is measured for three things: outdoor comfort, how hot or cool the courtyard feels; indoor daylight, how much natural light reaches the classrooms; and the energy use, how efficiently the building stays cool. The goal isn't one perfect shape, but a design playbook, a guide that says, for example, a courtyard with this ratio and that orientation can reduce heat stress, improve daylight, and cut cooling costs by like 20%. You may ask, why does this research matter? Well, because the physical design of schools can directly influence up to 16% of student learning progress. When children feel comfortable, their minds can focus on curiosity, not survival. This isn't just about architecture. It's about equity and ensuring that every child in desert cities can learn in spaces that inspire, not exhaust. My dream is that one day every school in Las Vegas and every higher city like it will use this playbook to design courtyards that turn heat into comfort and exhaustion into inspiration. Because when we design with climate and children in mind, we don't just build schools, we build hope that grows. Thank you.
Thank you so much, Elmira. Judges, you have 1 minute to score Elmira's presentation. Our next presenter, Oluwatobi Adeniji.
Hi, my name is Oluwatobi Adeniji, and my topic is automated treatment planning in radiation therapy. Cancer is continuous growth of cells and spread of cells in the body. It's an unwanted cell that we don't want. Radiation can be used to cure cancer. At the same time, it can cause cancer. We in radiation therapy, we are here to cure the patient. Just imagine you come into the treatment, they do your CT scan, they saw the volume of the cancer that you have. This cancer is not stopping, it's spreading. After 2 weeks, when your treatment is supposed to start, they discover that it has spread more than they expected. What causes that? It is as a result of delay in the treatment planning. Why do we have delay in treatment planning? It is because, imagine you have the tumor in the brain. You want to make sure that the radiation can always go into that tumor to stop the spread of that tumor. At the same time, you do not want it to go to the normal tissue. That is where the permutation comes in, where you try to give the high dose to the tumor, at the same time, low dose to the organ, so that those organs that are healthy, nothing will happen to them. Doing this trial and error, it takes time. How can we stop this longevity that always happen during this treatment planning? That is where I come in. I develop a module that will learn from the past treatment planning that they've done, pile it together, learn from it, use it to develop the new planning of a patient. Normally, manual planning takes 2 or 3 days. My module, if you put in just the SIM, the city of the patient, it's planned it within 5 minutes. With this, you can easily start the treatment the following day. This will prevent the spread of the tumor and also the spread of the tumor to other area. With that, you can easily treat the patient same day or the following day. These are the results of my module that I've created. We consider them. It is comparable to the normal manual planning that the dosimetrist or the physicist will do. And also, we have the graph that shows that this plan works very well with any site that we want to treat. So now we can easily treat the patient within a day or 2 days and will prevent the spread of the cancer to any other organs in the body. That's my project.
Thank you. Let's give us our judges 1 minute to score. Our next presenter, Megha Mohandas.
One word that dominate our headlines, our politics, and our planet's future: pollution. Right now, there are over 420 parts per million of carbon dioxide in our atmosphere, a level that's not seen in human history. This CO2 is a major culprit entering air through pollution, trapping heat, and driving global warming. And this is the challenge of our generation. Every single day, nature shows us the perfect solution. Think about a tiny leaf. A leaf takes sunlight, carbon dioxide, and water and converts it into food and fuel. And that's our dream, converting this pollution back into a valuable fuel using sunlight, the most abundant energy source that we have. Now, this dream is still down the line because CO2 is an extremely stable molecule and it takes a massive amount of energy to convert it into anything useful. Scientists are trying to achieve this conversion using special light absorbing materials, but these photo absorbers are associated with a critical weakness. When they absorb light and generate tiny charge carriers that need to run the reaction, they get easily recombined and fade away on the surface before CO2 can even react. And this inefficiency is a massive wall that blocking our path to a clean energy future. As in any challenging scenario, sometimes we need a team effort, we need a partner, we need a power couple. In my research, we introduced co-catalyst, which is a complementary material added to the surface of these photo absorbers. This co-catalyst is a perfect partner. It instantly grabs those tiny charge carriers, preventing it from recombining and speeding up the process. And it creates an entirely new junction on the surface and sometimes a completely new reaction site where the CO2 conversion can finally happen. I analyzed the surface of these catalysts atomically, observing exactly what happens at the junction of these materials during the reaction using highly surface sensitive techniques. It's all about narrowing down our fundamental knowledge so that we can effectively design the next generation and custom build highly efficient CO2 conversion catalyst. And this brings me back to the words of great visionary Dr. Buckminster Fuller. Pollution is nothing but the resources we are not harvesting. We allow them to disperse because we have been ignorant to their value. We believe it's time to being stop ignorant and start harvesting. Thank you.
Thank you, Mega. Now our judges have 60 seconds right now to provide their scores. Thank you. Our next presenter, Avinash Yaganapo.
Hello. Can you, yeah, hello. Hello everyone. Today I'm presenting one of my most recent work. It's a detection of senescence using holomatography images. Basically, senescence is the process of aging in which the cell stops divide and don't die. Traditional imaging technologies like bright field images, they have very low resolution images in which we can't see the internal organelles of a cell. So thanks to this new technology called holomatography, in which it captures a high resolution images, and we will be able to see the internal organelles, so that it will be easy to do detection for the senescence. So we developed a new model using contrast to learning, and we created a scoring systems for this senescence. And we compared our model with most of the current works, and we achieved like more performance than them, at least like 3 to 4% more than them. And in our model, we also developed a new loss function in which we compared that one with the traditional loss functions, and in which we were able to clearly separate the nucleus and the cytoplasm in a cell. And also we discovered some of the new biomarkers for the senescence, because in traditional imaging, as the resolution is low, it's other than the nucleus and the cell shape, we can't find any new other biomarkers. So from our method, like we found some new biomarkers in which like we can find like the solid substances inside a nucleus, and also like the cell membrane disturbing and also the white lipid droplets which get accumulated around the nucleus. So, and these are clinically proved as well, whatever these findings. So thank you.
Thank you, Avinash. Judges, you have 60 seconds to score Avinash's presentation. Judges, skip presenter number 8. Enter ones in your score, and let's right now turn attention to Dylan Brower. Dylan?
Hey y'all, thanks for being here. Okay, so throughout my time, I've learned that people's preliminary interactions with educators set a standard for them. I've seen that there's been a staggering decrease in students' interactions with classes, and I have dubbed this a social castration. There's many people in their late teens to early 20s who really struggle with things like ordering at a restaurant, or in our extreme cases, we see students can't even check out at grocery stores without freaking out. There's a plethora of reasons why, but I wanted to focus my efforts on understanding the social implications and what verbal and nonverbal rhetorical practices we can use to combat that social castration. I developed a methodology. I call it the 2 pillars of communication. These pillars are enrichment with outreach and education with representation. The way we so-called build these pillars is what I call my OCC method: outreach, connection, and communication. Firstly, we have outreach. That's the ability to understand an individual's strengths and weaknesses. I think of my Com 101 babies who sometimes they can't even introduce themselves without shaking. Research suggests that placing these students into collaborative and group work environments can help them break out of their shell and make a lot of friends. But this works especially well when we take those introverted students and place them in groups with the most extroverted students possible. Those extroverts thrive off of social energy so much that they can inject that into the introverts and help them crack out of their shell. But that doesn't always work. Then we move over to connection. As educators, this is what is learning what makes a student tick. What motivates them? What discourages them? Connecting with an individual either through nonverbal communication or social cues allows our students a path to grow and enrich their lives. Students who can't be broken out of outreach need instructors to give them that little push to get them out of their shell. Dr. Nick Tatum explains this very well in his research, which focuses on instructors using humor, specifically self-deprecating humor, to get those students to connect with you. More than anything, it makes them feel safe. Finally, we have communication, and that's the ways we interact with our students. What rhetorical strategies we as instructors use has a grave impact on them. The question is, how do individuals want to be represented? How can we better assist them to go be advocates for their communities? Working with Dr. Greg Hennigan on rhetorical strategies and effective communication is the foundation of my research. We, as instructors, all know that not every student can be pulled out of the cracks, but there are little tweaks that we can make in our classrooms to help some of our students from ever falling in. Thank you.
Thank you, Dylan. Judges, you have 60 seconds to provide your score. Thank you. Our next presenter, Declan West.
Hi everyone, my name is Declan and my dissertation is focused on how long giant trees can hold on to sugar for. One of my main species of focus in my dissertation is coast redwoods, which are the tallest tree species in the world, with the tallest individual being 380 feet tall. That's 30 feet taller than the Luxor Hotel on the Strip, which is amazingly huge. But why am I talking to you today about sugars? Well, as a quick reminder, trees make their own food in the form of sugars in their leaves. This gives them the energy that they need to survive. Trees' energy needs can be thought of as a recipe for baking something, such as a cake. When we bake a cake, we follow a recipe that requires a specific amount of each ingredient, such as flour, eggs, and crucially, sugar. When we want to measure out a specific amount of sugar, we use a measuring cup. And if we have any extra sugar, we generally scrape it off back into the bag. Trees do the exact same thing, except that this sugar is stored throughout the entirety of the tree. This sugar is produced during each growing season. But what happens if their energy needs change over time, especially in response to different disturbances, such as fungal infection or pest infection or wildfire? Well, we can track what season that this sugar was produced in using radiocarbon dating to get a comprehensive overview of when that sugar was produced and how they use that across time in response to different disturbances. The climate that redwoods evolved in is much different from our climate today. It is much warmer, fires are worse, there is much more fungal infection, there's much more insect infection. They've never seen this before. So getting a good comprehensive overview of how they use sugars in response to these different disturbances can help us better prioritize their conservation needs. Remember, if we need extra sugar for our cakes, we can go to the store to buy more of it. Trees, they can't. So if they have to rely on all of their stored sugar, what happens if they run out of it in time? We don't know, and that's what my dissertation seeks to find out. Thank you.
Thank you, Declan. Judges, you have 1 minute to score his presentation. And our next presenter, Catherine Obert.
I want everyone to close their eyes and imagine that they're taking a bite out of their favorite cookie. What does it taste like? Does it have chocolate in it? Is it chewy or is it soft or maybe even crunchy? In baking, bakers will often use a specific ratio of flour, butter, and sugar in order to achieve the taste and texture outcomes that they desire. For example, if you put too much flour into a cookie, it will come out dry and crumbly. Or on the flip end, if you put too little flour inside the cookie, then it will fall flat and become crispy. The human genome is a lot like baking a cookie. You can think of the DNA, which resides within each of us, as the recipe for life, and the nucleotide sequences that make up our DNA as the ingredients. Nucleotide sequences often occur in repeating patterns called tandem repeats, and this is quite common in our genome. However, when the ratio of those repeats double, triple, or even quadruple in size, that's when disease can occur. Many neurodegenerative diseases, such as Huntington's disease or muscular dystrophy, occur because of these tandem repeat mutations. So what does this have to do with autism spectrum disorder? Well, recently a study has been published that 36% of children born with myotonic dystrophy, which is a tandem repeat mutation disorder, are also diagnosed with autism spectrum disorder. And earlier this year, my lab made a key discovery that elucidates the molecular mechanism that connects these two. You see, what will happen is a certain RNA binding protein will bind to the exact sequence that is contained within this tandem repeat mutation. So when there's an increase in ratio of tandem repeats, There will also be an increased ratio of these proteins binding to that repeat. This means that these proteins are sequestered in the nuclease, and they are no longer able to perform their normal genetic function, nor are they able to interact with other genes that they usually interact with. These dysfunctional genes then go on to different gene expression and also form different protein isoforms that literally create a different type of brain. What we found is that these genes are also connected to autism risk genes. And currently, I am working on looking at individuals with different tandem repeat mutations in their genome to see if they also have a connection to autism spectrum disorder. So what I'm doing right now is I am using computational tools to analyze the genes that are affected by this mutation, and then using statistical analysis, comparing those genes to known autism risk genes. So far, the results are pretty promising. However, I still have a lot of work to do, but it is my hope that if this hypothesis rings true, then we are one step closer to unlocking the secret recipe that bakes an autism-flavored cookie. Thank you.
Thank you, Catherine. Judges, 1 minute to provide your scores for Catherine. Our next presenter, Ashley Roderick.
Imagine walking down the frozen aisle of your local grocery store. You open the glass door and you find Hidden Valley Ranch ice cream. Next to it, you find Kraft macaroni and cheese flavored ice cream. Because yes, these are real products, by the way. What makes one consumer choose these products instantly while another walks right on by? That simple decision of what makes a consumer embrace co-branded products while others don't is what my research aims to understand. Co-branding is when two brands come together to make a shared product or service. This approach has become increasingly common and increasingly complex across industries from fashion to food. At the same time, marketers of today have more access than ever to consumer data, offering new opportunities to make informed, data-driven decisions about how to target and engage their audiences. But here's the problem. Co-branding research still relies heavily on limited sampling. This means that our current understanding of how consumers react to co-branded products is based on a narrow slice of the population. Because of this, co-branding research tends to treat consumers as if they all act and think the same way. What's missing is the recognition that people bring in different identities, experiences, and backgrounds into every purchase decision that they make. My research brings an intersectional lens to co-branding research, and it puts the consumer lived experience at the center. Instead of viewing attitude and purchase intention as a straight line from awareness to buying, I argue that these interact circularly, consistently influencing each other based on who the consumer is and the context that they're in. By combining this intersectional marketing paradigm with existing co-branding theory, this new framework fulfills a critical gap, capturing real-world diversity in consumers that most models miss. Understanding why and with whom certain collaborations resonate can help save firms valuable resources and allow them to create products that people actually want to buy. In conclusion, my work looks to advance co-branding research into a new era, because when we understand the people behind the purchase, co-branding stops being a gamble for firms and can start being the innovative strategy it's meant to be. My name is Ashley Roderick. I'm an MBA candidate at the Lee Business School. Thank you for your time.
Thank you, Ashley. Ashley was our last presenter for today's morning session. I just would like to thank you all so much for joining us for our preliminary rounds. Thank you to the graduate college team who made this 3MT event possible. Now just stay tuned. We will announce winners live in just a few minutes. Judges, please be sure to press submit at the bottom of your scoring rubric form. While only the top 3 winners of this round will progress to the semifinals, you will all be winners of a Rebel Grad Slam t-shirt. You can get yours during the Rebel Grad Slam Finals reception. For the preliminary rounds, the first place winner will receive $300. The second place winner will receive $200. The third place winner will receive $100. And now just let's wait for the scores. Are we all ready? So here are the winners of this preliminary session. First place, Elmira Ebadi. Second place, Ashley Roderick. The third place goes to Catherine Oberg. Congratulations to all winners. And I would like to thank you again for your participation. And please, as a reminder, please join us for the semifinal rounds on Thursday, November 6th at 9 a.m., 11 a.m., and 1:30 p.m. in the Greenspan Auditorium. And the finals on Friday, November 7th at 4:00 p.m. in the Greenspan Auditorium. Lastly, we will have your Rebel Grad Slam t-shirt available at the finals reception in the Greenspan lobby. We welcome you to RSVP for the finals event, which is right now provided for you in the chat. And I just want to say thank you so much, judges and participants, for being here today. And I thank you so much for giving me the opportunity to be your host during this morning session. Thank you and congratulations.